Hello, everyone. Welcome to Old News, our monthly live stream about new discoveries in the field of paleontology. My name is Laura Beth. I am your host today, so I will be keeping an eye out on our live chat. And if you have any questions or comments, we would love to hear from you. Just type into the chat and I'll keep an eye on that. And if you would like to play bingo and learn some new vocabulary today, I just dropped a link in the chat to that bingo game so that you can play with us. And please, if you win, let us know in the chat. And I also want to let y'all know that if you would like to turn on closed captions for today, we've got some of those live automatic captions, and you just press the button that says CC at the bottom of the video. Now, I'm so excited to introduce our expert, Dr. Christian Kammerer, the museum's research curator of paleontology. Hey, Christian.
Hey, Laura Beth, great to be here.
I'm so excited for our topic today. But before we dive in, Christian, I wanted to give a little shout out to one of our participants,
All right.
Mallory. And Mallory has a birthday this week. So happy birthday, Mallory.
Oh, happy birthday.
Mallory wants to be a paleontologist, which I'm sure there
Awesome.
are some other. I know, right? I
Yeah.
know. And I think that actually, if you stick around for this program, anyone who wants to be a paleontologist, I think you're going to learn some really fun paleontology research. Going to learn about a new fossil discovery. I guess I don't want to give it away just yet. But yeah, we're so excited. Thank you so much for joining us today. And I'm going to go ahead and turn it over to Christian to share the
Yeah.
news.
So yeah, let's talk about the new discovery. So I feel like a lot of times on this show, we talk about new species of prehistoric animals that have been discovered. And those are great. Like a lot of the papers that are written are about sort of individual species. But this time we're talking about a whole new site, a new locality with a whole fossil assemblage there of lots and lots of fossils. And what's even better than that is it's what's called a Lagerstätte, which is something that we've actually talked about on this show a number of times, which roughly translates as sort of like the treasure trove of fossils. So let me pull up my screen here just to show you some classic examples of Lagerstätten. So these are sites that are sort of distinguished from normal fossil localities by the exceptional abundance and quality of preservation of the fossils. So you get lots of fossils, you get them in really excellent condition, very good preservation, and often preserved with things that you don't get normally, whether that be parts of soft tissue in invertebrates, or parts of the skin or hair or feathers, things other than the typical bone and shell that you get in most fossil preservation. And so here are just three examples of really famous Lagerstätten, all of which we've mentioned on the show in the past, like the Burgess Shale, which is from the Cambrian of Canada, right at the beginning of really the diversification of animals, the so-called Cambrian explosion, where we get all of these weird soft-bodied animals like Anomalocaris, this big, sort of weird shrimp, lobster-looking thing, but is a very distant relative of modern arthropods. Things like the Solnhofen limestone in Germany from the late Jurassic, most famous for having Archaeopteryx in it, but also this whole diverse assemblage of animals from this prehistoric lagoon, including lots and lots of fish and soft-bodied invertebrates. You get things like octopods and squid even from there, jellyfish, things that you wouldn't expect to fossilize otherwise. And then, of course, the Yehol biota from northeastern China, where all of these amazing feathered dinosaur fossils have come from. And there's also lots of fossils of early mammals and pterosaurs and early birds. And all of these have really, massively expanded our understanding of the prehistoric world and especially of prehistoric ecosystems. Because, even when you think about like the most abundant fossils, things like, microscopic algae or marine invertebrates, where the fossils can number in the tens of thousands or even millions, although, no one's actually going out there and counting every single diatom, but believe me, there's a lot out there. It's still a very biased record in the sense that, only certain parts of the organism are fossilized. So we're only getting like a shell or a bone or something like that. It doesn't give us basically the full complex relationships present in an ecosystem or a community. And for that, we really need to turn to these very rare Lagerstätten to understand what's going on. So we've talked about all of these Lagerstätten before, but there are also, and these are fairly ancient ones. So these are from the Paleozoic in the case of the Burgess Shale, or from the Mesozoic, the age of dinosaurs, in the case of Zollenhofen and Jehol. But there are also Lagerstätten that are more recent from the age of mammals, which also have a lot to tell us about what's going on in basically the origins of the modern world, if we want to think of it like that. So one example that is very well known from here in the United States is the Green River Formation in Utah and Wyoming. This is a lithographic limestone from a prehistoric lake, sometimes called Fossil Lake, that you can just see how incredible the preservation is here. This is an early horse. It's really every single bone preserved in articulation there. And so the Green River fossils, they are from the early part of the Cenozoic. They're from the Eocene epoch, around 50 million years ago. So, not quite, totally modern ecosystems. This is still, this is a pre-glacial world. This is near the Paleocene, Eocene thermal maximum. Earth is very warm at the time. The mammals that we know today are, the same groups are present, but they look very different from what we would expect today. Like this horse, for example, is only around the size of a small dog. It's not the big horses that we have today. And then you can also see it still has 5 toes on its feet, not the single hoof of a modern horse. When we look at the Green River Lagerstätte, it's dominated by fish fossils. There's just thousands and thousands of these things, especially, these little bony fish like Nidea and Diplomistis are incredibly abundant. I mean, these are some of the most common fossils that are found really in the US in general. And a lot of people who collect fossils probably have 1 or 2 of these on their mantelpiece. But because of the quality of the preservation, you also get these much rarer taxa, not necessarily, much rarer than some of these bony fish in life, but just because of the way they are preserved, you see them more rarely in the fossil records. So as an example of that, in the upper right there, you can see is a fossil stingray. So like rays and skates today, these have cartilaginous skeletons. Cartilage doesn't fossilize as well as bone. So we very rarely see these in the fossil record. There are a number of specimens of these early stingrays in Green River. There are also some bony fish, like in the bottom right. This Chrysopholis is a paddlefish, which is, it's an osteichthyon. It's a member of the bony fish group, but its skeleton is actually mostly cartilaginous if you look at paddlefish today. So this is also something that you don't usually see in the fossil record, even though we know they must have been there because it's paddlefish are a very ancient group. So because this is in a prehistoric lake, you're getting lots and lots of fish, but you also get really amazing preservation of the land animals and the air animals that are falling into the lake and being preserved. So at the left, this is a specimen that's on display at the Field Museum of a prehistoric lizard related to monitor lizards called Saniwa. And you can see both that it's really perfect from the tip of the nose to the end of the tail, like every bone is there. But it also has soft tissue preserved. Like if you look here, you can see some of the scales are preserved around the legs and around the base of the body. And then if we highlight the neck and the skull here, where I've marked with the red arrows, see those little ring-like structures? So that's actually the windpipe of this animal. So those are the individual cartilaginous rings of the trachea. So, and you can also see there's another fish preserved with it there. So just, incredible quality of some of these Cenozoic Lagerstätte. And this is really what's necessary to preserve certain taxa. So for example, on the right here, this is Acaronicterus. This is one of the earliest bats. And fossil bats are very rare because, bat bones are so fragile. They're small animals with hollow bones. So they have very, very low preservation potential. We have very few bats in the fossil record, especially complete skeletons of them like this. So really need this exceptional preservation in order to get them. So moving on from Green River, there's actually another site in Germany called the Messel Pit, which is also Eocene. It's around the same time. and also is sort of a lake ecosystem. So you're getting somewhat similar habitat there. Very different preservations. This is in an oil shale. It's thought these animals may have been killed by an eruption of maybe carbon dioxide or other toxic gases from the bottom of the lake. That's one of the hypotheses, at least, and may explain why we see the skeletons basically in such perfect shape, like they were all kind of, killed instantly and preserved as is. In some cases, this is actually another early horse. This one was preserved pregnant. So where I've highlighted in red here is where those tiny bones in its rib cage is actually the fetus of this extinct horse. So, it hasn't been scavenged or anything. It hasn't been pulled apart. It died and was basically covered in sediment almost instantly. And you see that with some of these other specimens there that they've got soft tissue preservation. Other mammals like Darwinius, which is an early primate on the left, you can see the fur covering there. And Copidodon on the right, you can see it had a big bushy tail like a squirrel. There's a member of a group called Cymolestins, which are, it's not a group that's alive today. It's a totally extinct mammalian order that you can sort of think of kind of weasel-like, but also kind of squirrel-like, probably mostly tree-dwelling. But like most of what we know about the anatomy of these things is coming from some of these Lagerstein sites. Some other animals that you get in Messel are also, very delicate boned things that are rarely preserved, like, complete birds like Messalornis here, or, some of the aquatic creatures like this small crocodilian that has every single scute preserved still in place. Of course, lots and lots of fish that were in the lake, but then also fossil behaviors are even being preserved there. So in these Elaeacheles, which is a prehistoric type of turtle, they were actually preserved while mating, which is one of the only examples of it, at least within vertebrates in their fossil record. So further evidence that these animals were killed basically instantly by something, whether that be sort of a gas explosion or toxic algal bloom that caused them to be covered in sediment basically as they were living. So all this is basically just to show that Lagerstein offer a really unprecedented and unparalleled view of paleo communities, really helping understand how these animals and plants lived rather than just sort of as isolated objects, and less guesswork for us as paleontologists trying to figure out what's going on, what they look like, which is really nice. So Green River and Messel, they're famous, incredibly productive, and incredibly informative. But they also, this is only a very small portion of the global land area of what's going on in the Cenozoics, and they're kind of at the same latitude too. So you've got something in Western North America, something in Europe. There was still connections between those continents at the time, and so you get kind of the same types of organisms on both sides. Which is why the new Lagerstätte that's been discovered is actually really exciting to me, because it's all the way down in Australia. So Australia does have a rich fossil record going back all the way to the Precambrian even, but is very well known for the incredible fossils from the age of mammals, especially giant marsupials and giant birds and huge relatives of the Komodo dragon and things like that. Australia is a very special ecosystem because it has been an island continent for so long. The fauna there is very different from what you get in the Northern Hemisphere. And we know that's been the case since the Mesozoic and even earlier for some groups of animals. So with an Australian Lagerstätte, you're getting really a completely different complement of organisms down there. And so the new site is called McGrath's Flat. It's in the eastern part of New South Wales in southeastern Australia. And it is a little bit younger than Green River and Messel. So it's in the Miocene, which is, not, it's in the Neogene, not quite as old as those Eocene ones, but also, not super recent. It's not like Ice Age. It's not within thousands of years. It's still millions of years ago. And so the animals that we're going to get in there are, in some ways more similar to what we have alive today, but it still just isn't like straight up the modern fauna. Let's take a look at some of the fossils from there, and they really are quite spectacularly preserved. So you can see one thing just is the color of the rock is this, it's a mineral called goethite, which is an iron oxide. So it has that really rich ochre color, that kind of orange that, is very typical actually in Australian rocks. Australia is sometimes nicknamed the sunburnt country. And if you go into the outback, you'll see a lot of these red, orange rock exposures. So it seems very, very typical of the country, these things. But also just like the preservation here, it really is incredible, especially of soft-bodied animals that otherwise have a bad fossil record, which is what we're really looking for in a Lagerstätte. So here's here's basically nearly complete spider, just missing a few of the legs on the right side, showing basically all of the anatomy. Still, all the animals from this site, work on it is really just starting. So none of these have been identified yet. Many of them likely represent new species. I mean, this animal should certainly be identifiable based on all the anatomy that's there. Insects, tend to be a little less soft-bodied than arachnids, and generally have a better fossil record, but still, their fossil record is mostly kind of isolated wings. So seeing, complete specimen like this is quite remarkable. This is a sawfly. Sawflies may be somewhat of an unfamiliar group. They might be called, sometimes they're thought of as primitive or early diverging, but they're relatives of bees and wasps. They're hymenopterans, actually quite diverse. And there's like 8,000 living species of sawflies, but for whatever reason, we think about them less than bees and wasps, maybe because they don't have stingers and they aren't always, posing a threat to us.
Yeah, that's gotta be it. Hey, Christian, I have a quick question about this picture.
Yeah, sure.
So, at the front of the sawfly's head, there are those two curved pieces. Are those antennae or are they like the mandibles?
So both are preserved in this specimen. So you can see how great the preservation is, because you can see here, if you look at the cursor, every individual segment of the antenna here is perfectly preserved. But then also the mandibles here, little slicing mandibles, also, just totally intact, articulated. So yeah, what you're looking at is you've got antenna there, right mandible, left mandible. This is the head in general. These would have been the eyes, and then thorax with the wings coming off.
That is incredible.
Yeah. Yeah, it's really, really remarkable. And it gets even better when you start looking at the fossils in detail. So those are things that you can just see with the naked eye that, oh yeah, it's a whole bug. That's great. But when you look at these fossils under a scanning electron microscope, you can see that even the microscopic structure of these things is preserved. And there are also microscopic fossils that couldn't be seen with the naked eye. So this is just from the paper by McCurry et al. Describing this site and introducing it. Some of these microscopic fossils, and to highlight a few, this is a fungal hypha. So hyphae, you can think of them kind of as the roots of molds and mushrooms and things like that. Getting into almost the cellular level of individual fungus cells, which you can also see in some of these yeast cells, yeast spores from a prehistoric fungus. So basically, some of them indistinguishable from yeasts today. And also in some slightly larger, but also very rare for fossils, things like fossilized nematodes. So nematodes are soft-bodied worms, and they are among the most abundant animals, and abundant and diverse of all organisms living today. There are nematodes everywhere. We think there are a lot of parasitic nematodes, including some of humans, but they're also super important, just free living in marine and soil ecosystems. And yet their fossil record is super bad because they're tiny and they're soft little wormy things, so they don't really preserve. So getting nematode fossils in this system also just amazes me. Even the larger animals, there are details of their anatomy that otherwise are not available in fossils. So if you look, this structure here that's sort of kind of oval shaped, this is actually an individual scale from the wing of a butterfly or a moth. You may have, may be aware that the wings of butterflies are made up of these individual scales, and if you touch one of them, which you shouldn't, but if you do, like all that little dust will come off. Those are the scales that actually make up the wings. So this is one of those scales, and you can see even the ultrastructure, the microscopic structure of what that scale looks like. And other tiny things like pollen grains from different fossil plants, which are very important for understanding, again, what the ecosystem was at the time. And like I said, this is a new site. It's just been introduced in the past few weeks. It's just been published on. So research is really only beginning, but it's clear that there's going to be years and years of new discoveries and sort of studies to be done on these fossils. So this is another set of figures from the paper, and just as some of the examples, like each of these could probably be a paper in and of itself of looking at microscopic scalation of some of these fossil fish. If this is part of a fish here, here's the head of that sawfly where they've looked inside microscopically and seen pollen grains to figure out what it was eating before it died. Looking into, this is a plant bug here that seems to have had parasitic worms in it, figuring out what was parasitizing it. Looking at fossil feathers and fossil leaves, like on the leaf. This is the stomata. So these are the little holes in leaves that plants use for gas exchange, for absorbing carbon dioxide and releasing oxygen. And stomata are actually very important for paleoecologists because the number, position, and sort of shape of the stomata actually tells you a lot about how wet prehistoric ecosystems were, like how oxygen rich the atmosphere was, all these sorts of things. So lots and lots of work to be done on this. And all very exciting. I guess the only downside is that there are, thus far, so few vertebrate fossils from there. I mean, it's mostly plants and insects. There are a number of these fish fossils, all seem to be one species. And so, we don't see any like cool, complete, marsupials or things like that. But when you look at, the proportions of fossils in other famous Lagerstätten, like Green River or Messel, these are the things that are usually found first, like lots and lots of fish, insects, leaves. And then much more rarely, and usually later after a lot of work, do they find something like a complete pregnant horse. So I hold out hope that there will be further discoveries of vertebrates in the future. And, again, vertebrates are only part of the story. Like most organisms are invertebrates and plants. Like we are, even with increasing human populations, we're still a minority of those things. With that said, in the Miocene in Australia is such an incredible time for the vertebrate fauna. When you think of things like these marsupial lions, these totally bizarre, like relatives of koalas and kangaroos that had become carnivores, some of these giant birds, the mihirungs, dramornithids that were present around then. These are known from actually good skeletal material from other sites, like Alcuda and in Riversley. So we have a good idea of what their skeletons look like. But to have one with the fur or the feathers intact, or to have gut contents to tell us what it was eating, I think would be amazing. So my fingers are crossed that new discoveries at McGrath's Flat bring some of these to the fore.
That is so cool, Christian. And I can tell you're really excited about those last two animals and the possibility of finding them. You're definitely gonna have to tell me more about that bird
Oh
because
yeah.
that was weird. But I don't know. I'm just amazed by this discovery for so many reasons. And it's super cool, like you said at the top of the program. It's really cool how we find, well, I don't find. It's cool how we discuss these discoveries, a new thing every month. And, we're usually finding it from like one fossil or a group of fossils. But it's just wild to me that after, at least 200 years of paleontology, we have
Mhm.
found a whole new fossil site, let alone a Lagerstätten. Did I say that
Mhm.
right? Yeah, that's like a treasure trove of fossils. Like that's wild to me. I don't know. Is it unexpected for you, a paleontologist as well, or is it?
Not yes and no. It is. It's not like in a wildly remote area, so that's a little surprising. It's close to, the most heavily populated parts of Australia. But just in terms of finding a new fossil locality, that is expected, because you never know, especially like what's going to be exposed at the surface at any given time. So one earthquake could reveal a new fossil site. One, new highway being built that has to cut through a hillside can reveal an entirely new fossil site. We're so limited in this science by, what is immediately visible when we have basically all of history buried underground. So there are doubtless like unbelievable Lagerstätten hundreds of feet or even miles, below the surface. Wow. That we may never see.
Man, but we may see them. The possibilities are endless.
Yeah. Well, I mean, there have been fossils, complete fossils found from cores from thousands of feet down.
Really?
Yep. So the only, for the most part, their coring that was done during geological exploration, a lot of it for like oil and gas. But sometimes they come up with really wild things. The only fossil from the age of dinosaurs of sort of a land animal from Florida, which was completely underwater at the time, like did not exist as a landmass during the age of dinosaurs. So there weren't any, T-Rex or anything walking around on Florida. It was just underwater. And all of the Cretaceous rock is very deep subsurface there, but there was a drill core that brought up a section of turtle that's in the Florida Museum. There's a complete little lizard-like creature called Boreapricea that was found thousands of feet below the surface in the Arctic Ocean by Russian scientists back in the 1970s. And so probably there's a whole assemblage, that's a Triassic animal, of these creatures down there, but good luck ever seeing any of that stuff again. That's really a 1 in a million chance.
Yeah. Well, that's incredible. And we had some great questions from Anne here in the chat.
All right.
So going back to that sawfly that I was asking you about, how big was that fly?
Oh, it's around the size of a regular bee.
Okay.
So like a honey, well, maybe more like bumblebee sized. So modern sawflies, some of them get pretty big. They get, they're around, some of them are among the biggest hymenopterans. So if you've ever seen like a giant hornet, around that size, but there are lots of smaller ones as well. So basically anything in the sort of typical bee range.
But I mean, the European hornet that we have around here in the summer, I mean, that thing is really big.
Yeah. And some sawflies, they get that big.
Yeah.
There's one, there's one group called the horntails in particular, that they really large, also sort of very, very bulky insects, very like slow flyers.
Okay. And Anne, I might butcher this pronunciation, but Anne wanted to know, are there any giant insects like Meganeura?
Oh, so those, I mean, Meganeura is one of the largest flying insects of all time. It's one of the giant dragonflies from the Carboniferous. But those really giant insects, they all went extinct after the Permo-Carboniferous period back in the Paleozoic. So nothing like that from these more modern assemblages. Like Cenozoic things, it's the same groups of invertebrates that we would see today, although probably different species, new species in these things.
Yeah, very cool. And then going back to how this fossil site was discovered, Anne wanted to know, was this discovery due to drought?
I don't think so. It's not like it was underwater or the area around it was, because that is happening. There are both exposures that were formerly subaqueous and places where the ground cover has kind of been killed off because of drought or extreme heat that has exposed the underlying rock. Just like a lot of fossil discoveries is there's a lot of rock out there and only so many paleontologists, and it's just kind of who happens to be there at the right time to identify this sort of stuff. With this particular site, it is the preservation, the exposures, my understanding is it's a little weird. So it's the whole area is covered in this ferrocrete, which is like iron rich it's kind of a breakdown of these iron oxide rocks that then are secondarily sedimented. And so they're getting chunks of the fossiliferous layer at the surface. So it's not, perfectly laminar like you get in Zollenhofen or any of these lithographic limestone places. So the actual fossil bearing layer is subsurface a bit, but some of it is being exposed at the surface there. So not necessarily something you would immediately just look at the landscape and say, oh, there's going to be an amazing Lagerstätte there.
Okay. And so we have a participant who got bingo.
Oh, wonderful.
Congratulations.
Congratulations.
To Martha for getting bingo. And I was wondering, so going back to, you were talking about the plants that they found, the fossilized plants. We haven't really talked about that on Old News, so I really don't know a whole lot about it. But you were talking about how the stomata can tell us a little bit about the climate conditions. So what did we find out about this fossil site? Like, what did it used to be like?
Well, it seems to have, certainly wetter than it is today. And with that said, it is similar to some, the fish that they're finding there is an animal called Retropinna, or at least a retropinid, something very closely related to Retropinna, it's a modern genus of fish that still lives in Australia. So we're looking at a maybe seasonally wet area. Some of the plants there are things kind of water lily-esque, so there is standing water at some times. It's not like full-on outback, just dry scrub desert. But it's also, it's not like it's deep water lagoon, anything like Zollenhofen, or even like Green River or Messel. So it's a more terrestrial sort of setup.
Yeah. It's so hard to imagine places as different than the way you know of them. So imagining Australia, I just picture the outback, the desert. And even if it's not a desert, I know they have forests and stuff, but I think they're more dry forests. So it's pretty hard for me to imagine that being covered in a wet kind of environment. But that's pretty cool. Do you think the paleontologists were surprised by that, or did they already know a little bit about how that environment was based on previous fossils?
Yeah, I think there was indications of that already. And in the Miocene in general, things are getting cooler and drier around the world. Like when you go back into the Paleocene and the Eocene, like there's a lot of like real jungle type habitats. And even like at that time, there are not tropical forests, but there are wet forests, even in places like Greenland. I mean, the world was not glaciated at the time. And then as you're getting into the Miocene and the Pliocene, increasingly a colder, drier world. But again, it's not, universal across the globe. So, I mean, you're seeing the origin of grasslands in a lot of places like North and South America, but then there are also tropical forest belts where there aren't today, deserts in some places where there are forests today. And, it's the complexity of changes in where continents are, ocean currents, and global temperatures is very difficult to suss out. And so it's something that, is like paleoclimate is such an active field of research. And it's still, at some sites, we still even don't know kind of what the temperature was. We have to rely on a series of proxies of, what's going on with the stomata, what's going on with oxygen isotopes, things like that, in order to infer these. But it's hard to be sure. Just look at one fossil site and say, oh, okay, it was exactly this hot, and we got, X centimeters of rain every year and all these things. It's basically impossible.
Yeah. Oh, well, that's unfortunate. But I mean, every bit of information is helpful. So
yeah, for sure.
And so we've talked on Old News before, I think, about how Australia is kind of known for its dangerous animals, right? So are there any dangerous animals that we can find in these fossils that we know of that we'd be able to tell, like, oh, that spider?
Well, yeah, I mean, we don't know how exactly how venomous that particular spider was. I guess, it's for invertebrates, that is almost impossible to determine, I would say.
Even with
Like,
these fossils?
I mean, but the thing is, all spiders are venomous, so you know it's venomous. With
Mhm.
fossils of this quality, you can probably see, you can see the fangs. You may even be able to see with something like, you know, uh, scanning tunneling microscopes or CT scanning or something, even be able to reconstruct where the venom sacs are. But you won't be able to tell the toxicity of it from that.
Right.
Uh, you can tell again, you can't tell the toxicity. So in venomous vertebrates, you can tell how much venom they were capable of transmitting in a bite based on venom gland size and sort of like diameter of, like in a cobra or something, or a rattlesnake, diameter of the venom grooves in the teeth. So there are, it's possible to do inferences there, but one, we don't have any of those at this site, and two, it's just the chemistry of the toxins is probably not surviving.
Yeah, so those chemicals would probably have degraded over
Yeah, for sure.
years.
They
For sure.
degrade
Okay.
quickly in the modern world. It's one of the reasons why creating anti-venins is such a problematic issue, because you need to be able to get fresh venom in order to create the anti-venins.
Oh, okay. Yeah, I didn't know that, but that, I mean, it makes sense.
Yeah, I mean, they have other ways now, but they used to just like, we're constantly milking snakes and then injecting the venom into horses and then waiting for the horses not to die and take their blood, which then had the antivenins in it, and then they'd inject that into humans to deal with snake bite.
Wow. How long ago was this?
Oh, this is not that long ago. I feel like this is still in my lifetime, at least. They were injecting venom into horses to make antivenoms.
Okay. Wow. And what about colors of the fossils? You mentioned how these are really colorful fossils, which
Mm-hmm.
I'm sure is from the Goethite. Is that how
Yeah,
you pronounce it?
Goethite, named
Goethite.
after Johann
Okay.
Wolfgang Goethe, the German polymath and novelist.
Okay. So that's giving the fossils a little bit of the color, but I know that when we get into when we get microscopes in paleontology,
Mm-hmm.
we can
Mm-hmm.
start to see or understand what colors things were. So can we do that with these fossils? Have they done any of that?
I think they're already starting. So here, let me just bring this back up. So up here, I think these are parts of feathers, and they're looking in B there at these melanosomes, and that's what was used to figure out the colors of the Chinese feathered dinosaurs. So that sort of work is being done there. And again, this is very I don't even want to say controversial, but it's a lot of debate among paleontologists how rigorous that is and how true that is to the actual color versus how much of that has been altered by bacterial degradation and things like that. But they're clearly giving it a shot. And then some of the other ones, because it's against that orange background, you're losing a lot of the natural color. Like this leaf here it would have been a green leaf when it was alive. It's not red, but we should be able to see like patterning on some of these things, like if they had stripes or spots or things like that.
Yeah, that still is really impressive to me. And I have one last question for you, Christian.
Yeah.
Out of all of these fossils that they found at this one site, what's your favorite or most surprising find that you are most excited about?
I think, well, I mean, there seem to be multiple nematodes, but yeah, probably the nematodes, both because they're one of the things that have the worst fossil record, but they're one of the things that are most important to modern ecosystems. And so especially having some record of maybe both free living and parasitic nematodes in this environment, it's not really something that we've seen in many other fossil assemblages at all. So there are some sites that yield amber that have nematodes that are like coming out of insects and things. Nematodes or nematomorphs, some of them. But again, this is just kind of scratching the surface of this. I'm sure as they start, also because I mean, you're not going to see this with the naked eye. Like these are things that you're going to have to bulk sample lots of this goethite exposure under the microscope before you figure out just how many nematodes are in there. And could be a lot, could really teach us a lot about what's going on with them.
How do they tell the species, like species of different nematodes.
Oh, with these fossils, you're not going to be able to, I think.
Okay.
Even with the quality of preservation here, unless you can see fine details of their sort of like the hook-like appendages that are in sort of their various orifices, you're not going to be able to tell them. And some of them might be high enough resolution to do that, but like the one that I showed before that had like part of its body eroded off from where the rock was broken, you're not going to be able to identify that just beyond nematode. Even like with modern ones, a lot of the classification is based on genetics now rather than on external morphology. So it's not something we can do with the fossils.
Right. But just knowing that they were there is valuable information.
Yeah, and like higher level identification, it will be possible, like not down to the species level. But like I said, nematodes are a hugely diverse group, and there are like many different types of nematodes. They aren't all just the same thing. They aren't all C. elegans.
Yeah, that would be the one that I know of the most. C. elegans. Is that the one that's parasitic? Is that why I know about it?
No, that's the one that it's sometimes called the worm, as if there's only one, because it's the major study system in genetics and developmental biology. It's the worm used in labs, basically. So
okay, great.
And it's free living. Like parasitic nematodes you're probably familiar with are things like pinworms or roundworms, things like Ascaris, which is among the biggest nematodes, which gets in your gut and can be like, as thick as a finger. It's horrible stuff to have Ascaris in you.
No thanks.
Yeah.
I'm good. All right, everyone. Well, that is all the time we have today for Old News. Thank you so much for joining today and playing bingo and asking great questions. We hope that you can join us again next month. Mark your calendars for February 8th, 1 p.m. Eastern Time. And if you'd like to receive a reminder email or materials for playing Old News bingo, bingo definitions, and any extra resources, you can register at the link in the video description. Christian, as usual, thank you so much for sharing your expertise and helping us understand what this new discovery actually means for us.
Oh, my pleasure. So excited to be able to talk about this stuff.
Yeah. Well, have a great day, everyone. We'll see you next month.
Yeah, see you next month. Stay safe out there.
Oh, yeah. Bye.
Bye.